Hi everyone, in this video I'm going to explain what is next generation genome sequencing. So before understanding the next generation genome sequencing, we should know what is the first generation sequencing and second generation sequencing. We all know that the Sanger sequencing is a first generation sequencing where we fragment the DNA and then we sequence these DNA fragments one after the other. But in second generation sequencing, where also we fragment the DNA. After fragmentation of DNA, we read or we sequence these fragments simultaneously. So since we do this simultaneously, so it reduces the time. So not only the time, it also reduces the cost of sequencing. So now let us go to the next generation sequencing. How many steps are there and what these steps are. So first step is library preparation. So in library preparation, so the DNA is fragmented. So DNA which is collected from the sample is fragmented by using some methods like restriction enzymes, or it can be done by using ultrasonication also. So after DNA is fragmented into pieces, so these pieces are then joined with the adapter sequences or the adapter fragments. So here also you can see the first step is library preparation, in which the DNA is fragmented into pieces, small pieces. Then these small fragments are joined with the adapter sequences you could see here with the red color and cyan color. Okay, this is the first step. So after this, the sample which has these small fragments is then loaded onto the uh, machine, so which then perform the second step. So the second step is sequencing the library. So first we have fragmented DNA, so this fragmented DNA pool is called a library now. So then this library is sequenced by the sequencing machines. Okay, that is the second step. So after sequencing of these small fragments, okay, we get the reads. So then these reads, so okay, then these reads are mapped to the reference genome. When these reads are mapped to the reference genome, how we could get more reads? See, when we load this library onto this machine, so this machine performs PCR amplification. So that means each fragment is sequenced multiple times. Why each fragment of DNA is to be amplified multiple times? When we have multiple pieces for a DNA, you know that DNA polymerases are not they don't have high fidelity. So that means they make some errors when they prepare a complementary strand. So if we sequence only one time, so what happens when we sequence only one time? So maybe the fragment which is synthesized in this step may have some mismatches. So these mismatches can be eliminated when we have a multiple number of when we perform multiple rounds of amplification. So when we have multiple copies for a target DNA, so then we can eliminate the bases which are not common among all the reads. Or we can say, suppose we have 10 reads. Out of these 10 reads, suppose we have 3 reads which have mismatch with the reference genome, so we can eliminate these three reads which are showing the mismatch, and we can confirm that okay, these three mismatches are due to the PCR bias, or we can say that that is because of the fault while amplifying the particular reason during the PCR during the PCR reaction. Okay, so this is the second step where we prepare multiple copies for a given fragment, and these multiple copies are called reads. And these reads are two types, short reads and long reads. It depends on the machine that we are using. Some machines they produce very short reads, that means up to 150 nucleotides only some machines they can produce. Then some machines they can produce even 1000 base pairs, some can produce even 2000 base pairs, like that. And coming to these short reads, okay, 150 nucleotides, since uh, that reads have 150 nucleotides, that means so that PCR is performed for 150 cycles. That means in each cycle, so one nucleotide is added to the that growing complementary strands. That's why 150 times or 150 cycles is performed to complete or to synthesize the short reads. Okay, this is the second step. The third step is the alignment part. This is also called as the data analysis part. So three three steps are there. First one is library preparation, second one is the sequencing the library, third one is the data analysis. So for this data analysis we need to use some computational methods to align these reads with the reference genome. So we have reference genomes for organisms, and the NCBI has the NCBI, on NCBI we can find reference genomes. So we could get the reference genome sequence from NCBI, and then we can match the our reads with the this reference genome. Suppose there is some there if suppose some changes are observed in the reads with respect to the reference genome. Suppose A is present in our reads and T is present in the reference genome. That means there is a mismatch between the reads and the reference genome, and this mismatch we call it variance or variation. So that means in the sample there is a variation with respect to the reference genome. So that means the reference genome sequence is completely different from the sample sequence. So when there is a variation, so we can go and check what this variation and which gene that variation, and what is the effect of this variation in the function of that particular gene and all. So the basic purpose of doing this next generation genome sequencing is to find out the variation among the population. That is the reason, and also to find out the insertions and deletions. And see, when we go to the analysis, there are two things which are very important. One is depth and other one is coverage. When you see this this is the reference genome. When we map this our reads with this reference genome, we could see that see the entire this genome has the reads. So when you see the I will show you some other image. So I will show you other image where you could see that some sequence, some area of the reference genome doesn't have any sequences. So for example, you could see here this part which is highlighted by in a gray color. There you could see you don't find any reads against this reference genome. So that reason we call it no coverage area. So in some areas where you could see more number of reads which are more number of reads against the reference genome. So when we have more number of reads for a particular reason, we can say that is a we could say that is a highly covered area. And when we have less number of reads for a particular area, for example on the right side next to right side to this gray area where you could see that 2 reads present, okay, and left to this gray area where you could find 3 reads are present in this particular area. So that means some areas where you see high coverage, some areas you see no coverage, and some areas where you could see less coverage. Okay, this is very important. So coverage is this one, and the next one is depth. That means depth is means how many reads see once we know the coverage, so you could look into see these reads. See, these all these reads are not in the similar size or similar length, I could say. See, when you map these reads against the genome, you could see that, see, if suppose first you could start from the 5 prime end. So suppose a nucleotide is present in all the reads, you can say that means no, this area has a high depth. We could say high depth. When you go to the 3 prime end, where you could see only one read has the 3 prime end, suppose there is a nucleotide T, you could say that this area of the genome has a less depth. So depth is in terms of nucleotides present on the reads the against the reference genome. And because of this, because the reads which are not similar in length, so we could see the difference of the depth in the same reads itself. That means the same coverage area, you could see difference of the depth. Okay, that is very important. And coming to the coverage, why we don't find reads which are reads for particular region of the reference genome? Why? Because you know that the genome DNA contains some repetitive sequences. So when this region is uh, sequenced, so that region produces multiple copies. That region produces reads like any other area of the genomic DNA. But when this software they try to map this particular reads to this reference genome, since this repetitive DNA is present in the present at several places in the DNA, so the softwares cannot they cannot they cannot map this to a particular region. So because they find multiple reasons where these reads could be mapped. So that's why there is a lot of confusion, there is a lot of problem to map these um, reads to the genomic reference genome. That's why we could find some reasons where we don't find coverage. So those reads are eliminated by the softwares because these reads cannot be mapped to a particular place. That's why see we find some areas with very less having less coverage, or we can find some areas without also coverage. So this is about the next generation sequencing. 3 steps are there. So for the third steps, that is called data analysis, which requires requires computational methods to analyze the data. So in our upcoming videos there I will discuss how to analyze the data. Okay, thank you so much.